Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss purine nucleotide metabolism. In the last video, we discussed cell division and its association with cancer. Before a cell divides, the chromosomes have to be copied so that the daughter cell will also get its 23 pairs of chromosomes. And that chromosome copying or DNA synthesis happens here in the S phase of the cell cycle or the synthesis phase of the cell cycle. We shall discuss DNA synthesis or DNA replication in a future video. Before the DNA can be synthesized, before the chromosomes are copied, the cell needs to make all the substrates for DNA polymerase. This is the enzyme that will copy DNA. And these substrates for DNA polymerase include the deoxyribonucleotides, the purine nucleotides, as well as the pyrimidine nucleotides. So we shall look at this synthesis of purine and pyrimidine nucleotides first before we talk about DNA synthesis. And that's what we'll do in this video as well as the next video. Just a quick reminder about the structure of a nucleotide. It consists of the nucleobase, which could be either purine rings, guanine and adenine, or pyrimidine rings. For DNA, that would be thymine and cytosine, and uracil is something you find in RNA. And then we have the sugar. In case of DNA synthesis, this will be a deoxyribose. And then we have the phosphate groups, the three phosphate groups. So copying DNA by DNA polymerase will need deoxy-ATP, deoxy-GTP, deoxy-CTP, deoxy-TTP. So we'll look at how these two, the purine nucleotides, are synthesized in this video. And then the next video, we'll look at how these two, the pyrimidine nucleotides, are synthesized. So this figure here gives you an overview of the of nucleotide metabolism. So on the right-hand side here, shown in this blue box, is the metabolism of purine nucleotides, which we'll focus on in today's video. We shall only look at reactions and enzymes that have medical relevance, that have connections, especially with pathology and pharmacology. You can find details of these pathways in biochemistry textbooks as well as on the internet. We'll see how purine nucleotide metabolism is associated with genetic diseases such as the Lesch-Nyhan syndrome and SCID, how it is associated with cancer, how purine nucleotide metabolism is associated with immunosuppression, and also how it is associated with a disease called gout, as well as how purine nucleotide metabolism is important for chemotherapy drugs as well as for immunosuppressive drugs. Now, in order to make the purine ring, the raw materials that are necessary include glycine, the amino acid glycine, the vitamin folic acid in the form of tetrahydrofolate, the amino acid aspartic acid, the amino acid glutamine, as well as carbon dioxide, which sometimes is written as bicarbonate. So whenever you see CO2 or H2O3, it's basically the same thing because carbon dioxide dissolves in water to give you carbonic acid, which then breaks down to give you bicarbonate and protons. The sugar part of the nucleotide comes from ribose 5-phosphate, which, if you remember, mostly comes from the hexose monophosphate shunt. The phosphate groups of the nucleotides come from ribose 5-phosphate as well as ATP. Now, there are two pathways to synthesize purine nucleotides. One, shown here is the blue arrow, is called de novo synthesis, which means synthesis from the beginning, from scratch. And then we have the salvage pathway. And in this pathway, the nucleobases are simply recycled. So let's first look at the de novo synthesis of purine nucleotides. In the first reaction, we see the formation of a reactive ribose sugar. This is called 5-phosphoribosyl pyrophosphate, or PRPP. In this reaction, ribose 5-phosphate reacts with ATP to form PRPP and AMP, and this reaction is catalyzed by this enzyme called ribose phosphate pyrophosphokinases, also called phosphoribosyl pyrophosphate synthetase. So when we say active or reactive sugar, what we mean is you can see this carbon here of the ribose right here, carbon number 1, has a nice group, a pyrophosphate group attached to it. In organic chemistry, this means this is a good leaving group, and that will make this carbon number 1 reactive so that it can undergo the next reaction where you can see an amine now attached to carbon number 1. This enzyme is inhibited by some of the products of this pathway, especially ADP and GDP. So ADP and GDP, which are some of the products formed in this pathway, when they accumulate, they can inhibit this enzyme called ribose phosphate pyrophosphokinases. In the next reaction, you can see an amine group that comes from the amino acid glutamine is attached to the reactive carbon, carbon number 1 of PRPP. So basically this reaction is PRPP plus glutamine plus water as the substrates, and the products are 5-phosphoribosylamine plus glutamic acid plus pyrophosphate or PPI. And this reaction is catalyzed by this enzyme called amidophosphoribosyl transferase. This is the regulatory step of the purine synthesis pathway. This enzyme is inhibited by some of the end products of the pathway, such as AMP and GMP, as well as their diphosphates and their triphosphates, such as ADP, ATP, GDP, or GTP, as well as XMP, one of the intermediates that is formed in the pathway. PRPP, which is the substrate for this enzyme, is actually an activator. So the more PRPP that is present, the more this enzyme will be active, and this is called feed-forward activation. So here we had feedback inhibition, where the products will inhibit the enzyme. Here we have a substrate activating the enzyme. That's called feed-forward activation. Then in the next few steps, the 2 rings of the purine ring are now assembled step by step on the activated sugar PRPP. Now, the synthesis of these purine rings requires 3 amino acids, glycine, glutamine, as well as aspartic acid. It also requires a 1-carbon chemical called a formyl group, and this formyl group actually comes from the amino acid serine, and it's carried by this vitamin called folic acid in the form of tetrahydrofolate, THF. It also requires carbon dioxide or bicarbonate. So you can see CO2 is not just a waste product, it's actually used as substrates in several reactions in metabolism. So after these series of reactions, we form the first complete nucleotide, and this is called inosine monophosphate, or IMP, and the nuclear base here that you find in IMP is called hypoxanthine. So the first purine nucleotide the cell makes is IMP. Now once IMP is made, now it can be converted into the GMP nucleotide or the AMP nucleotide. In the formation of GMP, what happens is the IMP is first converted into XMP, and this is done by the enzyme IMP dehydrogenase or IMPDH. In the next reaction, the XMP that is formed by the IMPDH reaction is converted into GMP by the enzyme GMP synthetase. So these two reactions will convert IMP into GMP. Now GMP is a competitive inhibitor of IMPDH. So this is another example here of feedback inhibition, because GMP is a product and it's coming back to inhibit an enzyme early on in the pathway. Notice that for making GMP, we require ATP. ATP is used. Okay, later we'll see that to make AMP, we'll use GTP. The cell uses GTP. So this is done because it wants to balance the concentrations of these two purine nucleotides, the A as well as the G. Now let's look at a connection with pharmacology, and that involves this enzyme IMP dehydrogenase, which makes IMP into XMP, and then that XMP is converted into GMP. There are drugs that can inhibit IMPDH, and these drugs are often used as immunosuppressants. So for example, the drug mycophenolic acid, it's a non-competitive inhibitor of IMPDH. These immunosuppressant drugs are used to prevent organ rejection and also for treating certain autoimmune diseases. The lymphocytes, the T and the B lymphocytes that are involved in organ rejection as well as autoimmune diseases, these cells rely very heavily on the de novo synthesis pathway to make GMP. So by suppressing GMP synthesis, the cell would not be able to make, eventually to make deoxy-GTP, and without this nucleotide, the chromosomes cannot be copied, and these lymphocytes cannot divide and proliferate. And without lymphocyte proliferation, they would not be able to reject the transplanted organ, and they would not be able to destroy the patient's own tissues in autoimmune diseases. Not surprisingly, these drugs were also tried against cancer as chemotherapy drugs, but what we see so far is there is limited efficacy against cancer. And the side effects of these drugs, since the drugs affect other rapidly dividing cells, for example, red blood cells, we see anemia in patients taking the drug. Then GI cells also divide very rapidly, and their proliferation will be affected, and that leads to symptoms such as nausea, vomiting, as well as diarrhea. Since these drugs suppress the immune system, so patients taking these immunosuppressants are at very high risk of infections. That's a serious side effect of these drugs. Okay, so we have seen how IMP is converted to GMP. Now let's look at how IMP is converted to AMP. So first, IMP is converted into adenylosuccinate, and this is carried out by the enzyme adenylosuccinate synthetase, and then the adenylosuccinate is eventually converted into AMP and fumarate, and this is done by the enzyme adenylosuccinate lyase. We see another feedback inhibition since AMP, the product AMP, will inhibit this first step here, the synthesis of adenylosuccinate. So this is an example of feedback inhibition. So feedback inhibition is a very common way to regulate the amount of a certain intermediate. So if the concentration of AMP, as it increases and increases, more and more will go and inhibit this enzyme and therefore slow down the synthesis of AMP. So a very powerful way to regulate the amount of intermediates or products in the cell. So as mentioned before, in order to balance the amounts of AMP and GMP, so here in the synthesis of AMP, we see GTP is used. GTP is used for the synthesis of AMP. Earlier we saw how in the synthesis of GMP, we actually use ATP. As we shall see in a few minutes, these two enzymes here are important parts of what we call the purine nucleotide cycle. So we'll discuss the purine nucleotide cycle in a few minutes. So once the AMP and GMP is formed, it can be converted into its diphosphate. So GMP can be made to GDP, AMP can be made into ADP, and this is carried out by the enzyme nucleoside monophosphate kinases. These NMP kinases can use nucleotide substrates where the sugar is either ribose or deoxyribose. In the next step, the enzyme ribonucleotide reductase will convert the GDP into the deoxy form, deoxy-GDP, and the ADP into deoxy-ADP. Ribonucleotide reductase requires the protein thioredoxin to reduce the enzyme that becomes oxidized when it catalyzes this reaction. Now, not surprisingly, this enzyme is under very tight regulation in order to maintain the correct balances of all the various deoxynucleotides. ATP is an activator of this enzyme, while deoxy-ATP is a feedback inhibitor of ribonucleotide reductase, and we'll see this becomes an important point later in a disease. In the next step, enzymes called nucleoside diphosphate kinases or NDB kinases, they will convert all the diphosphates into triphosphates. So all the diphosphates, whether they have ribose sugars or deoxyribose sugars, they are all converted into their triphosphates. Of course, GTP and ATP are used for RNA synthesis, while the deoxy forms are used for DNA synthesis. So there we have it, synthesis of the deoxypurine nucleotides that are needed for DNA synthesis. We started from here, the activated sugar, and went through these steps, and now we have the substrates necessary for the enzyme DNA polymerase in order to synthesize DNA, in order to copy the chromosomes. Now let's look at the catabolism, the breakdown of purine nucleotides. Nucleic acids are broken down during several processes. During digestion, we see RNA and DNA will be broken down in the small intestine. RNA is broken down all the time in cells. DNA is broken down mostly. Some parts of DNA are broken during repair. And of course, when the cell dies, then there is a breakdown of DNA as well as RNA. So enzymes called nucleases, example deoxyribonuclease and ribonuclease, these enzymes that are found in cells as well as pancreatic juice, for example also in lysosomes in cells, these enzymes basically will hydrolyze the phosphodiester bonds. So remember, the one sugar of a nucleotide is connected to the other sugar of a nucleotide by phosphodiester, and these enzymes will break down the nucleic acids into smaller fragments, oligonucleotides, and eventually into nucleotides. So during this breakdown, once the nucleotides are formed, the phosphates can be removed to form nucleosides. And this is catalyzed by enzymes called 5' nucleotidase, as well as various phosphatases such as alkaline phosphatase. So various nucleotides can be converted to nucleosides. So for example, AMP can be made into adenosine, IMP into inosine, XMP into xanthosine, and GMP into the nucleoside guanosine. The nucleosides that are formed can then be broken down into nucleobases, and this can be done by breaking the N-glycosidic bond between the nucleobase and the sugar. This is carried out by enzymes called purine nucleoside phosphorylases, or PNP, as well as enzymes called nucleosidases. So inosine can be converted to hypoxanthine, the nucleoside xanthosine can be converted to xanthine, and the nucleoside guanosine can be converted to guanine. Now during digestion, most of the bases that we ingest in our diet, they are mostly degraded and excreted in the GI system. So we rarely absorb these bases into our bloodstream. Now here we mention inosine, xanthosine, as well as guanosine, but we did not mention adenosine. So let's look at adenosine breakdown, as well as a reaction of another reaction by which AMP is metabolized. So there is an enzyme called AMP deaminase that can make AMP into IMP, releasing ammonia. Later we'll see this is another enzyme that is involved in the purine nucleotide cycle. Now adenosine as well as deoxyadenosine, these are not broken down by PNP. Okay, so adenosine and the deoxyadenosine doesn't go through this reaction. adenosine is broken down by another enzyme called adenosine deaminase. Okay, so don't get these two confused. This is AMP deaminase. This is adenosine deaminase, also called ADA. And what happens is adenosine is hydrolyzed to give you inosine and ammonia by this enzyme ADA. And once we get inosine, now the inosine can be broken down by PMP into hypoxanthine and the sugar. Now, in the disease called SCID, or severe combined immunodeficiency disease, there is mutations that cause defective ADA. Okay, so this enzyme is activity is impaired, and that leads to problems with lymphocyte functions in these patients. The lymphocytes do not work. So what seems to be happening in these patients is in the absence of ADA, deoxyadenosine, which is also broken down by ADA, accumulates. And this deoxyadenosine is phosphorylated to form DATP. This deoxyadenosine phosphorylation is very predominant in lymphocytes. So now we have very high concentrations of deoxy-ATP. Now this high concentration of deoxy-ATP inhibits ribonucleotide reductase. We saw that earlier in this step. Deoxy-ATP is a powerful inhibitor, feedback inhibitor of ribonucleotide reductase. So this causes this inhibition of ribonucleotide reductase means the lymphocyte cannot make the deoxynucleotides, especially deoxy-GTP. That means the nucleotide cannot copy its chromosomes before it divides, so the nucleotides will not, the lymphocytes will not be able to proliferate, and therefore the immune system will be compromised. So these patients will have a problem dealing with infections. So SCID patients are very sensitive to infections. The 1976 movie that was called The Boy in the Plastic Bubble was a story about a boy, David Vedder, who had SCID. Okay, before we continue with purine nucleotide catabolism, let's say a few words about the purine nucleotide cycle. This was something we mentioned in video 5.29 when we were talking about starvation. We looked at or talked about this green part here, which was part of the purine nucleotide cycle. So this cycle involves these three enzymes that we talked about: AMP deaminase, adenyl-succinate synthetase, adenyl-succinate lyase. And when you add up these reactions catalyzed by these enzymes, cancel out all the common intermediates such as IMP and AMP and so on, you end up with a net equation, and that is basically this amino acid is converted into fumarate. Okay, so aspartate is converted into fumarate. The purine nucleotide cycle has a lot of physiological functions, some of which we are still studying. One of its connections is with exercise in the skeletal muscle. So during exercise, of course, the skeletal muscle needs a lot of ATP, and fumarate that is formed this fumarate that is formed can enter the mitochondria, go through the Krebs cycle, and then eventually be used to generate energy. And so this energy generation seems to be important because in the disease AMP deaminase deficiency, in this genetic disease AMP deaminase deficiency, some of these patients, not all, but some of the patients actually experience pain and cramping during exercise. So it's possible that one of the functions of the purine nucleotide cycle is to provide energy to the muscle, especially during exercise. Another important function of the purine nucleotide cycle is the generation of ammonia. So this cycle helps regulate intracellular nitrogen. It removes excess ammonia. So the exercising muscle will send that ammonia to the kidneys in the form of glutamine. And in the kidneys, the ammonia is required to neutralize the excess acid that is produced during exercise. Okay, so acidosis that happens during exercise, what we call exercise acidosis, the ammonia helps neutralize the protons that are generated during exercise. Okay, back to catabolism. Okay, so as we saw earlier, the enzyme PNP will break down the nucleosides into the nucleobases such as hypoxanthine and guanine. Now, in the next step, the enzyme xanthine oxidase will convert hypoxanthine into xanthine, while the enzyme guanine deaminase will convert guanine into xanthine. The xanthine that is formed is then converted into uric acid by the enzyme xanthine oxidase. So you see that here in this reaction, xanthine oxidase converts xanthine into uric acid. Now the uric acid is secreted into the bloodstream, and then it enters the kidneys and is excreted into the urine. Now, the enzyme xanthine oxidase is an enzyme that is known to reduce oxygen into superoxide or hydrogen peroxide. Now, these two chemicals, superoxide as well as hydrogen peroxide, these are part of the reactive oxygen species. There are many reactive oxygen species. These are two of them. And these ROS, reactive oxygen species, have been associated with many diseases. They cause what we call oxidative stress. And we see this problem in cardiovascular disease, in reperfusion injury. That means when there's reoxygenation after ischemia. We see ROS involved in several neurodegenerative disorders. So reactive oxygen species are involved in many diseases, and you can see that xanthine oxidase might be associated with ROS. A few words about uric acid. Remember that the nitrogen of amino acids of proteins is removed in the urine as urea, but the purine nucleotide nitrogen is removed as uric acid. Note that the purine ring is still intact in uric acid. So humans cannot break down the purine rings into simpler chemicals. As we mentioned earlier, the importance of reactive oxygen species in diseases, it's interesting to note that uric acid has antioxidant, that means it can scavenge free radicals, which is good, but it also has pro-oxidant effects. That means it can actually produce free radicals, which is bad. So uric acid has both good as well as bad effects when it comes to oxidative stress. Now, uric acid is a common, routine clinical chemistry test. Elevation of serum uric acid can happen in various conditions. If there is too much purines that is broken down, for example, in Lesch-Nyhan syndrome, then this purine breakdown will lead to formation of uric acid. In addition, if there's any problems with the kidneys cannot excrete the uric acid. Some uric acid is also excreted by the GI tract. If there's a problem there, that's going to increase the serum uric acid. Now, the problem with elevation of serum uric acid is that uric acid is already very poorly water soluble, and even slight increases in the uric acid may cause the uric acid to precipitate. And once it precipitates, it can lead to inflammation and even cause kidney stone formation. And this condition where there is excess uric acid which precipitates, this condition that leads to inflammation, this is called gout. This disease is called gout. For treatment of gout, there is a drug called allopurinol. This drug inhibits xanthine oxidase. So without xanthine oxidase, hypoxanthine is not converted to uric acid, and xanthine is not converted to uric acid. So now instead of uric acid, we see more xanthine and hypoxanthine. Now these two chemicals are much more water soluble than uric acid. Therefore, they are less likely to precipitate and cause inflammation or kidney stones that we see in gout. Okay, so we have discussed the synthesis of nucleotides as well as the catabolism of nucleotides. Now let's look at this part here called the salvage pathway. Now in the salvage pathway, there is this enzyme called hypoxanthine guanine phosphoribosyl transferase, or HGPRT, and this enzyme can react the nucleobase guanine with PRPP to give you a nucleotide, GMP. Similarly, it can react the nucleobase hypoxanthine with PRPP and you get this nucleotide IMP, which can then be converted into AMP. So basically in this pathway, we see the nucleobase being converted directly into nucleotides in one step. If there is any free adenine nucleobase present, which is not usually the case, because as we saw, the adenosine is catabolized into hypoxanthine, okay, instead of adenine. So if there is some adenine present, even that adenine can react with PRPP to give you AMP, and this is catalyzed by the enzyme adenine phosphoribosyl transferase, or APRT. So basically in the salvage pathway, we see that the bases, the nucleobases, are converted into nucleotides by reaction with PRPP. So what's the importance of this salvage pathway? Well, purine synthesis from scratch, de novo purine synthesis, is quite expensive in terms of energy. We are talking about 8 to 9 ATPs required to form AMP or GMP. So this recycling of purine bases by the salvage pathway is, in terms of energy, it's an economic strategy to provide nucleotides for the cell. Now, red blood cells do not have the de novo pathway, so they rely completely on the salvage pathway for their purine nucleotides. And by recycling the nucleobases, the cell can prevent the formation of excess uric acid. As we saw, this excess uric acid, because of its pure water solubility in the bloodstream, it can precipitate and lead to gout as well as kidney stones. So there is a very important genetic disease called Lesch-Nyhan syndrome. In this disease, the enzyme of the salvage pathway HGPRT is defective. This gene for HGPRT is present on the X chromosome, and therefore this disease affects mostly males because they have only one X chromosome. So this is an X-linked recessive disorder. So since the purine bases are not recycled, they will accumulate, and that will lead to their catabolism into uric acid. So in these patients with Lesch-Nyhan syndrome, we see increased or elevated serum uric acid, and as mentioned earlier, that can lead to precipitation in various parts of the body, leading to inflammation and the disease gout. And in the kidneys, this uric acid can precipitate and form stones. Now, Lesch-Nyhan syndrome is associated with several neurological problems, including intellectual disability, problem with movements, as well as muscle stiffness. However, the one characteristic that seems to be very strange that is seen in these children with Lesch-Nyhan syndrome is this self-mutilation behavior where they bite their own lips and fingers and they bang their heads, and that exact reason for why this unusual behavior is not known, but it may affect some sort of dopamine signaling in the brain. Okay, so that's all for purine nucleotide metabolism. In the next video, we'll look at pyrimidine nucleotide metabolism and related medical relevance. Once again, thank you for supporting the channel. Have a wonderful day. We'll hopefully see you soon.